Good, thanks everybody for being here. My name is Brandon Cooper, and I'm going to talk to you today about our work on Wolbachia and the Drosophila yakuba complex of flies. So Wolbachia are the most prevalent symbiote on the planet. They're alpha proteobacteria that most arthropods and also most insects have. We think about 60% of arthropods have them. I think that's probably a lower bound because we score these with PCR, so those don't always pick up low titer infections. They're passed on by mom vertically, so they should co-evolve towards mutualism. More recently, they've become quite known as serving as a biocontrol of vector-borne disease. So you may have seen these in the news as you were preparing to come to Austin. You've heard about Zika. When you take Wolbachia from Drosophila melanogaster and you put them into Aedes aegypti, they block the transmission of arboviruses like Zika and dengue. Finally, some of them seem to be rare, but in Drosophila, in a couple of systems, we see that Wolbachia seem to contribute to reproductive isolation. For example, in the Polystorum complex, they have obligate relationships with Wolbachia and hybrids. The titer, the number of cells the hybrids have is messed up, leads to hybrid inviability. And then in the system that Kelly's going to talk about, Drosophila subquaternion and sister species reasons, Wolbachia seem to have contributed to reinforcement. I say here Wolbachia are master manipulators because they became quite well known for manipulating post-reproduction by killing males. Another trait is this cytoplasmic incompatibility trait that I'll talk about today. So if you have an infected line and an uninfected line, and you complete all possible crosses there, you'll see one of those crosses, the one between uninfected females and infected males, leads to a reduction in egg patch. Importantly, that the cross where females also have Wolbachia, the eye-by-eye crossing, can be rescued. We don't totally understand how this works, but it's interesting because Wolbachia modify the sperm goes out into mom, and then somehow an infection in the females can rescue whatever Wolbachia has done to the sperm. Now, I've been working in the Drosophila yakuba clade, which is 3 hybridizing species. They're found in Africa. The sister species Drosophila yakuba and Santa Mea co-occur on this island, Sao Tome, off the west coast of Africa. Santa Mea is endemic to that island. Yakuba occurs throughout continental Africa. The outgroup to those sister species, Drosophila tseri, that emerged a little over 2 million years ago, occurs on another island, Bioko, and also throughout continental Africa. So on the island of Bioko, you have tseri and yakuba, and on the island of Sao Tome, you have Santa Mea and yakuba. Now we know they both have Wolbachia, but I'm going to show you some sort of in-depth analysis of what those Wolbachia are doing. We know that these post-hybridize, and this makes this an interesting system for thinking about. Remember, I told you that Wolbachia are in 60% of arthropods and also most insects. This is a nice group to think about how Wolbachia might spread via introgression, because in a couple of generations you can get spread from Wolbachia from one species to another. Imagine Santa Mea, female mates to a yakuba male. Santa Mea has Wolbachia, that F1 has Wolbachia, and then in a one generation back cross with the yakuba, you can get Santa Mea, Wolbachia, and mitochondria into yakuba. And so it's an interesting system to think about cytoplasmic here. And so the questions we had is how much genomic variation is there for Wolbachia in this group? What do they do to the host? If they cause CI, do they cause, and that's that cytoplasmic incompatibility effect, if they cause that, do they cause interspecific CI that could contribute to post-zygotic isolation? And do they have any effects on mating within or between species? So the first thing we looked at is we have Daniel Matuti shared, and he's a collaborator on this, Illumina data, whole genome Illumina data, where we were able to pull out Wolbachia genomes and ask the question, how much genomic variation is there, and whether or not these species have their own Wolbachia? What we found is there's not a lot of variation, about 114 SNPs, but 13% of that variation, only 13% of that variation is shared among the host species. So if you find your favorite SNP in a Wolbachia, in Santa Mea Wolbachia, you pull that out, odds are you're only going to find it in Santa Mea. Now we know that these hybridize historically and to a certain extent in current time. You'll notice that this phylogeny recapitulates the host phylogeny. Now that's not evidence of this being an older infection. We're trying to figure out what's going on, and I'll show you some fitness effects in a minute that we think might be maintaining the limited Wolbachia variation there is within each host species in the face of introgression. So do these Wolbachia manipulate host reproduction? So we conducted large-scale experiments to look for cytoplasmic incompatibility. And what we find is they do. So what you'll see here on the x-axis are all 3 species first, and for each of those 3 species we have all 4 possible crosses. The first thing I want you to notice is the 3 low bars within each of those classes. That's the cross between uninfected females and infected males. So what you're seeing here is a reduction in egg to adult viability based on the Wolbachia in that one side of a reciprocal cross. And we have all 4 crosses here, so the next thing I want you to see is that the I-by-I crosses, we also see the females are rescuing this effect. So we have cytoplasmic incompatibility in this group. It seems to be between 20 and 30%, and females can recover that trait, or rescue viability. In Drosophila tseri, we notice this effect where it seems to be segregating variation in CI intensity. So if you ask a number of lines, they may differ. I'm just showing you 2 examples here, where in one we have no statistically significant cytoplasmic incompatibility, and in another we do. Now remember, I showed you we have all this genomic variation, so we were interested in, does this variant, this Wolbachia control this difference in CI intensity? And so the way we got at that is that we introgress these Wolbachia, I'm just showing you this, one of the same figures I showed you before, into common backgrounds, and then we look to see, does this persist? So in most Wolbachia systems, you would put the Wolbachia in a different background, and you would see it still does or does not cause cytoplasmic incompatibility. And so we did this, and what we see is that when you put it into a different host background, cytoplasmic incompatibility can actually go away in some cases. So the host seems to retain some control over this. You can think about it as host suppression to a certain extent, stopping this manipulation of reproduction by Wolbachia, on the host. We can take one that doesn't cause statistically significant CI, and we can put it in another background and ask the question, can we turn this into a Wolbachia that manipulates host reproduction? And in fact, we do. So when we put this in another background, this Wolbachia starts to look like the ones that we see that cause, in their natural background, a 20 to 30% reduction in egg to adult viability. And this is a cat trait in this case. So these are all intraspecific effects. The next thing we wanted to know is, do they cause these effects interspecifically? Because as I showed you, they hybridize. It's been shown that they hybridize in the lab, and we know they hybridize in the field, and so we want to know if these effects occur across species boundaries. And what we see is, so first what I'm showing you here are just the reciprocal crosses. So I'm not showing you all 4. The first thing I'll tell you is that females do rescue cytoplasmic incompatibility, but to make this figure a little easier, I'm only showing you the reciprocal crosses. On the x-axis, you have all of the possible interspecific crosses. So for example, the first 2 you see are Santa Mae and tseri. And what you can see is that the blue bars are always lower. And again, this is that cross between uninfected females and infected males. So we see interspecific CI in these crosses between species that diverge. The sister species about 1 million years, and the outgroup crosses to tseri closer to 2.5 million years ago. So we have interspecific cytoplasmic incompatibility. So a question you might have is how might this matter for post-zygotic isolation? And so one thing that I presented last year that I'm not going to show in depth today is that on these African islands we've been tracking through time, there's also fluctuations in the frequency of Wolbachia. So remember that the I-by-I, the infected by infected crosses, females can rescue this. What we see is that in 2015, infections were at about 80% in yakuba, and they had decreased in Santa Mae down to about 30%. So what this means is on average, if you cross a Santa Mae female to a yakuba male, that's going to be the sort of cross that leads to cytoplasmic incompatibility. On the other hand, if you cross a yakuba female to a Santa Mae male, that cross is most likely not going to cause cytoplasmic incompatibility. So this is a pattern that could lead to asymmetry in female rescue that could contribute to post-zygotic isolation, but I don't think that we can say much about that yet, because these fluctuating frequencies that lead us to say this, also suggest that these conditions may not persist. So if we come back and track these infections 5 years from now, we may see, that Santa Mae has come back up. And so that's going to be a question we're going to have to answer through time. The next thing we wanted to know is, do these Wolbachia generate assortative mating within species or pre-mating isolation between species? So I've shown you that there's this pervasive intraspecific and interspecific cytoplasmic incompatibility. So a question might be, okay, well maybe it doesn't matter, Maybe females can sort of sniff out an infected male. I'm an uninfected female, no, I'm not going to mate with an infected male, because if I do, I'm going to have a 20-25% reduction in egg hatch rates. So what we did is we set up all the possible intraspecific crosses, all the possible interspecific crosses, 100 males, 100 females in vials. We come back in 24 hours. We dissect all those females to see if they've been inseminated. And when we do that, what I'm showing you here are the within species crosses, and what you can see is a pattern here of females discriminating against infected males when they don't have Wolbachia would be the blue bars being lower. And we don't see that here. So regardless of Wolbachia infection status, these females are mating at the same rate with males, regardless of their infection status within species. Now when we cross these, we do the between species crosses. Remember, Drosophila Santa Mae and yakuba, those are the sister species. that diverged about 1 million years ago, and TSE is the outgroup to that sister pair that diverged about 2.5 million years ago, we think. And what we see there is, again, a similar pattern of Wolbachia not mattering or not impacting mating behavior. So the first thing you'll see is the very low bars on the x-axis for the Santomea by TSE cross on the far left, and the yakuba by TSE cross on the far right. That's just because these are so diverged. They just don't mate very regularly in that period of 24 hours, but it doesn't have anything to do with Wolbachia. For the sister species pairs, just look at those pairs of bars, and you can see that, again, the blue bars are not lower. Those are reciprocal crosses again. So the cross between uninfected females and infected males, uninfected females are not discriminating against infected males. They're still mating with them at the same rate, given this assay. So if we come back to these questions we had, we wanted to know how much genomic variation is there among these Wolbachia that infect different hosts. We see that there's limited variation, right? There's about 100 SNPs or so using these Illumina data. We're currently getting PacBio genomes to figure out what these variants are that we see, but only about 13% of the SNPs are shared among Wolbachia from different hosts. So they do seem, I mean, these are not different strains, but the Wolbachia variation we do have seems to be partitioned within hosts, and that phylogeny currently recapitulates the host phylogeny despite being, we think, quite recent infections. Yakuba complex Wolbachia manipulate reproduction in the form of cytoplasmic incompatibility within all 3 of the species, and they also do so in the between species crosses. We do see those asymmetries in female protection that we could say, you know, could possibly contribute to post-zygotic reproductive isolation, but because that's based on these fluctuating frequencies that seem to be going up and down through time, and I didn't show you also vary through space, we think we need to track them longer to see. I think what we're going to find is that Wolbachia don't matter in the long term for patterns of post-zygotic reproductive isolation. And then we see that they don't matter for mating behavior, so females presumably are experiencing this in nature because they're not discriminating against infected males. They are mating with them both within and between species, so I'd like to do some field experiments to see what, you know, if we can actually pick up CI in the field. With that, I'd like to thank Michael Torelli and Daniel Matuti, my main collaborators here, Will Connor, I've been working with on phylogenetics and genomics. Paul Ginsberg is a great tech in our lab that's getting ready to go work with Kelly. And David Turasini in Daniel's lab has also been working with us on the genomic portion of this research. I'd like to thank the NIH for my funding and for Torelli's funding. I'm getting ready to move to Montana, so if you have any good undergrads that want to be techs or grad students, let them know if they're interested in Wolbachia. We have a good evolutionary genetics group there. And with that, I'll take any questions.